This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 32, recorded April 26th, 2012. Hello, everyone. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from San Diego, California, Elio Schaechter.
Hi there. How are you?
How are you, Elio?
Good.
I have a feeling I haven't seen you in months. Is that true?
It's not quite, but it's about a month.
A month.
I'm glad we're back.
And happy birthday.
Thank you very much.
I'm glad that you were
105.
110. Good to have you again. Also joining us today from the Medical University of South Carolina, Michael Schmidt.
I, too, send my birthday greetings to our colleague in San Diego.
Thank you.
And welcome our guest panelists today that you're going to introduce.
We have a special guest today. We don't have too many guests on TWiM, but today we do have one, and she is a professor of zoology at the University of British Columbia in Vancouver, Rosie Redfield. Welcome.
Hi, and happy birthday, Elio.
Thank you so much.
Whenever it was. Is it today?
Yeah, thank you.
It's today.
Yeah.
It's amazing. Huh? We're having a recording TWiM, and it's Elio's birthday. What better present, right?
Absolutely. Can't beat it.
So, Rosie, it's okay to call you Rosie, right?
Yes, that's what you should call me.
Excellent. And I got your affiliation correct, right?
You did, yes.
Have you been at UBC for a long time?
I've been here for 22 years.
Was that your first position out of a postdoc?
I was here I am a microbiologist, and I often am sort of incorrectly connected to the microbiology department. My first job was here, soft money, in the biochemistry department. And then I got a position, hard money, permanent position in the zoology department about 20 years ago.
Ah. So you were trained as a microbiologist, though?
Well, no. Well, no. I did my PhD on phage lambda, so I guess that makes me a microbiologist, even though I've never taken a microbiology course.
That doesn't disqualify you.
Right. So I was trained by Alan Campbell. I think that makes me a trained microbiologist.
Oh, you were with Alan Campbell?
Yeah.
Wonderful.
Wow.
And then with Ham Smith at Johns Hopkins.
Oh, this is fabulous.
So those are definitely microbiologists who trained me. So
yeah, excellent. Yeah, excellent training. Wow.
I met Alan Campbell at a meeting in Los Angeles a few years ago, and he was just a joy to talk with.
Well, he's still going strong. I saw him last summer. I had lunch with Alan and his wife, Alice. They're both still working in the lab.
That's right.
Wow.
Alan and I were Army buddies. We were together at Walter Reed Army Institute of Research. That's a long time ago.
Wow.
And, of course, Ham Smith is working like crazy in the lab for Craig Venter.
Great. So what we're going to do today, we want to talk about a paper published in Science some time ago called a bacterium that can grow on arsenic instead of phosphorus. We've had a number of requests from listeners to discuss this paper, and we've never done that on TWiM. And in addition, Rosie, we've had people request that you come on and talk about it with us. So here you are.
Well, here I am.
So we thought we would go over that paper because we haven't, and then we know that from your blog that you have been doing experiments, and we'd like to talk with you about those as well.
All right,
so like a plan.
Good.
So maybe you could start by telling us what was done in that original Science paper.
Okay, how do you want the 10-second version or the 5-minute version? Or
I think a 5-minute version. We might interrupt you along the way.
You can interrupt me. So I first heard of it, I just started using Twitter, and there were these tweets about NASA calling a special press conference to talk about possibility of interstellar life or something like that. And then there was, in sort of science blogging, I saw people talking about this paper in Science, reporting that they had discovered bacteria that could replace the phosphate in their DNA with arsenate. And I was already pretty skeptical of NASA's attempts at microbiology, because I was around when they claimed to have found evidence for life in that Mars meteorite, of which the sole evidence was they had found tiny little things that were sort of shaped like E. coli, but 10 times smaller, and that looked kind of like little bits of crud that a geologist had called nanobacteria. So I was skeptical. And when I read the paper, I became much more skeptical, because the paper was full of flaws. What the researchers had done was they'd taken sediment, so a tiny bit of scientific background. As best I can sort of infer, partly from what they said in the paper and partly from other background, the goal of the research was to find evidence of what's called the shadow biosphere. This is an entirely hypothetical but very cool kind of organisms that might have, the idea is we want to study how life originates, but right now we only have one example, because all of the living organisms we know of are descended from the same common ancestor. But maybe back when life was getting started, more than one form of life evolved independently, and maybe one of those forms of life has survived and is living amongst us unsuspected, because we wouldn't recognize them as life because they're not the kind of life we know. So that's the shadow biosphere hypothesis. And this research was supported by NASA in the hope of finding organisms with different chemistry than all the organisms we know. So the premise was maybe there were organisms that could use arsenic instead of phosphorus, in the same way that science fiction stories talk about space aliens that are based on silicon chemistry instead of carbon chemistry. So that's the motivation, as best I can understand it. What the researchers did was they took sediment from California's Mono Lake, which has a very high arsenic content, about 200 micromolar, and put that sediment into a simple defined culture medium, basically high salts, pH 9.8, which, because Mono Lake's very alkaline, glucose, vitamins and minerals, but no phosphate was added to the medium. Instead, a whole lot of arsenate was added to the medium, initially around 1 millimolar, but then up to 40 millimolar. And it was very exciting for the researchers, because something grew in that medium that looked like bacteria. As the paper describes it, they characterized the growth of these organisms, which look like bacteria, in medium with or without arsenic, with or without added phosphorus. And the organisms grew very well with arsenic. They didn't grow very well without arsenic, at least when there wasn't any added phosphorus. They sort of collected the cells and analyzed them in a number of ways. Some very simple and familiar to most microbiologists. They extracted the DNA and tested the DNA for arsenic and found lots of arsenic, well, some arsenic at least, in the DNA. And they also tested the wash bacteria in various ways for how much arsenic was there and what kind of chemical bonds it appeared to be forming, using a lot of very high-tech molecular, nano, micro, mass spectrometry stuff that I don't actually know anything about. And from that work, they concluded that these bacteria were actually incorporating arsenic into their biomolecules with the same kinds of bonds that phosphate would have, and in particular that the bacteria had incorporated arsenic into their DNA. So that was the gist of the paper.
And they chose Mono Lake initially because it has a lot of arsenic in it. Is that correct?
It has a lot of arsenic. It also has quite a bit of phosphate. It's 200 micromolar arsenate, 400 micromolar phosphate. It's also very hostile to most forms of life because it's high salts, about 1 molar salt, and very alkaline.
There's some little flies that live there. In fact, the Indians used to harvest them and eat them, eat the larvae. So they do well, and brine shrimp does well there too.
Yeah, so it's not an extraordinarily high level of arsenate. Anyway, that was where they chose to look, and they found these organisms. But one other thing they found when they were characterizing these organisms was that they weren't part of the shadow biosphere, because one of the researchers on the paper sequenced the 16S ribosomal RNA of these organisms, 16S ribosomal RNA gene. So first that tells us they have DNA, and they have DNA that Taq polymerase and sequencing machinery can process. And that sequence showed that these were, in fact, not shadow biosphere, but they were halomonas bacteria, part of a fairly well-studied group.
Well, let me ask, this is a direct digression off of that paper, but in thinking about the shadow biosphere, I'm always perplexed, why use glucose? If you think about the early life of planets, I wouldn't anticipate we would necessarily have that much glucose around.
At the start of life, indeed.
At the start of life, and I would have thought that they would have, if they're thinking about a carbon source as the principal substrate, they would have gone to a 2-carbon unit like acetate or, carbon monoxide or something along those lines, rather than glucose, because it goes back to how you design your enrichment. Because if you're doing a shadow biosphere, you want to enrich for organisms in that shadow biosphere, and it really is principally along the lines of, what you're hunting for. And since, to the best of my understanding, phosphate is never used as an energy source. You don't.
Right. It doesn't really have any energy. Yeah.
Yeah. It's never used as an energy source. So I was, when I looked at the paper the first time, I was perplexed about that. I could understand the substitution of phosphorus and arsenic or arsenate, but then when the chemistry, when I had my flashbacks to my qualifying exam of thinking about arsenate and some of the other issues that I'm going to bring up a little later, it troubled me. And so the shadow biosphere is a very attractive concept, but I think their original experimental design / enrichment was a little bit flawed if you took classic enrichment biology that most graduate students are trained with, or undergraduates are trained with, if they do indeed elect a major in microbiology.
Right, and maybe because I was not properly trained in microbiology, that didn't immediately strike me as a problem, although in retrospect, the medium was kind of weird because it had every vitamin known to be needed by modern organisms, and a complete set of trace metals, and then glucose. And yes, that was a little naive, maybe. In any case, something grew. Their tests said that there was arsenic in the DNA. NASA was thrilled. They had this press conference, massive media coverage, the authors sitting in a panel showing a video with arsenic, I guess they were arsenate molecules going into DNA in place of phosphate. It looked really great for about 2 days. Even then, even on the panel, they had Steve Benner there, who was being, the devil's advocate and saying, well, gee, I don't know. And most of the mainstream media coverage was fairly, they believed what the paper said. They believed what NASA said. The science blogs were a little more cautious. They always ended with, well, this is really cool if it's true. And I didn't get to read the paper for a couple of days, but when I read it, I was really appalled at the many, many flaws and inconsistencies and red flags in the paper. One really big one was that they presented 2 kinds of growth curves, one where they counted cells and one where they just measured the optical density, and they were quite inconsistent. And they looked superficially the same until you realized that one was on a log scale and one was on a linear scale. Then you went, wait, in that case, they shouldn't look the same. And it was not at all clear from reading the paper, but on thinking about it afterwards, people realized that the cells had divided for a little while, then they'd stopped dividing, but they kept getting bigger because they were accumulating probably poly-beta hydroxybutyrate granules, which is a carbon energy storage material that bacteria accumulate. And then that would also explain something that the authors mostly referred to as just vesicles, which was these big white globs in their electron micrographs of the cells. So there was this big red flag about, wait, these two growth curves aren't even consistent at all. Even though the error bars on them were really tight, the two versions didn't make any sense.
Yeah.
And the numbers it turned out one of the first things you realized when you read the paper, and then especially when you looked at their supplementary materials, was that the medium that they used had a lot of contaminating phosphate in it. Although it was hard to be sure because different batches of their medium had different amounts of contamination, and they hadn't kept track of which, or at least they hadn't reported which batches were used. But it looked like, on average, their medium had about 3 or 4 micromolar phosphate in it. Their no-phosphate medium had that much phosphate in it. And they, in the paper, which had an unfortunate kind of salesman-y, I'm trying to sell you something tone to it, in their paper they argued that wasn't nearly enough phosphate to explain the growth that they saw in this medium. But in fact, when I did a sort of a back-of-the-envelope calculation saying, well, say they put 10% of their phosphate into DNA, it was just the right amount of phosphate to explain how much growth they got. So the bacteria didn't need, as far as I could see, the bacteria probably didn't need to use arsenic to grow. They had enough phosphate to grow to the levels that were seen. But the worst problem was in their analysis of the arsenic in the DNA. They had gone through what they claimed was a DNA purification. They'd isolated DNA from the cells and run it in a gel and then analyzed the bands for phosphate and arsenate. But, and this was just shocking, they had not purified the DNA away from the gel. So what they analyzed was the actual gel slices, for which at least 99.9% of the biomass would come from the agarose and the gel buffer, not from the DNA. So when they found arsenic in their DNA, purified DNA preps, there was no way to tell whether that arsenic came from the gel, came from the buffer, because the DNA hadn't been carefully purified before it was loaded on the gel. It was phenol extracted and chloroform extracted to get rid of things like proteins and lipids. But all of the soluble salts would have remained with the DNA. It had been put through one ethanol precipitation. They later said they had washed the pellet, although that wasn't mentioned in the paper, and then just loaded directly on the gel. So there could have been a lot of arsenic carried over from the medium and the cells into the gel as well. So it was very unconvincing, their argument that the DNA was actually contained arsenate. So what happened was, I read this paper, I said, oh, this is terrible. And I wrote a blog post. I have a research blog in which I mostly write about day-to-day about my own experiments. And I've been doing that for about 5 years. But sometimes I'd write about other papers that I'd read. And so I thought, okay, I'm going to blog about this paper. I didn't really expect anybody to read it because nobody really reads my blog. At least nobody
You sure?
did. I mean, even my students had to be kind of nudged into reading my blog, even though it was all about our research. But by writing it down in a blog, I knew that I would, A, clarify my own thinking, and I would, basically get my ideas straight on paper. And then I'd also have something I could refer people to when I got into arguments with them about this paper. I could say, look, I've explained it all on my blog. Just go read it. That was my intent. So I wrote this blog where it was as if I was reviewing the paper for publication, but with not being quite as circumspect in what I said as I would be if, they'd submitted it and I was a reviewer. So I think the harshest thing I said was, I don't know if these researchers are bad scientists or if they've just been swept away by NASA's enthusiasm for extrastellar life, which was true. I don't. Oh, and I also said, if this work had been brought by a graduate student to their committee meeting, I would send them back to the bench to do more cleanup and more controls.
Well, it even starts as simply as how you graph logarithmic growth curve, optical density-based growth curve data. I was always taught that if you're doing a growth curve, you always plot it on a semi-log plot with optical density on a log scale and time on a linear scale so that you may easily determine the generation time.
Right.
And in the science paper, they clearly did not do it on a semi-log plot.
No, their optical density is on a linear scale.
And they're trying to argue about growth. And that would, going through the editorial process, I just didn't understand how the editors or the reviewers actually missed that really basic point that we teach in introductory microbiology. If you're trying to argue about growth, and these authors were truly trying to argue about growth, you should at least use the commonly accepted standard convention of plotting it on a semi-log plot in order to see what's going on.
You just hit a very raw spot with me. It turns out that there are paper after paper after paper that has never heard of what you correctly say is what is taught early in the game. I see more papers where growth is described on a linear scale, and it makes no sense because you cannot look at it and tell what the growth rate is. In fact, most people who talk about the growth rate have no idea how one measures growth rate. Growth rate is a growth rate in a certain medium for a certain bug. That's it. That's what happens when bugs are growing in exponential growth in an unhampered condition. They're not using up critical material. They're not accumulating toxins. Under those conditions, it's a single growth rate, and you do this by studying cultures at a fairly low density. Okay, this is not. I could go on for an hour. I'm sorry. This is really.
Yeah, sorry. I could too, because this was. After I read this, I said this would be a good exam question for a Micro 101 course.
Oh yeah, absolutely. This is the classic way of understand. If you really understand the basic population structure and population dynamic of bacteria, and it's an extremely important point. I'm going to put a link to a paper that I found doing a quick search while we were talking about this. It's in the public domain. It's called Theory and Measurement of Bacterial Growth by Friedrich Weidel. And it's basic and practical aspects. And this was published in 2007 with a correction in 2010. And it's from a German journal. But it's a really good place that sums up the theory and practical applications. It describes OD. It describes how you graph it. Describes the mathematical model. So this will help those interested in the audience in getting this right.
So, Rosie, what was the response to your blog post?
Well, it might have, not been seen by anybody, but I did post comments on a couple of blogs about it. Blogs that had discussed this paper. I said, bottom line, dreadfully, shamefully bad, something or other. And I did send out a tweet about it, but at that point I only had one Twitter follower, who was Jonathan Eisen, and who I know didn't see it till the next day. But luckily there were a few people who did read my blog, including a former postdoc who sent out a tweet and had more followers. And so that by that evening, I had hundreds of people looking at my blog, which was great. And by the next day, I had like 10,000 people looking at my blog, which was thrilling beyond words.
Yeah, that's when I stumbled upon you, because I was reading about the paper somewhere, and they referenced, you have to go read Rosie's blog post. So that's what I did.
Right. And that's what a lot of people did. And it was what I found particularly rewarding was that I was getting comments from good scientists and tweets from scientists, people whose judgment I respected, saying, go read Rosie's blog. She's got it right. Mm-hmm. Now, at the same time, I have to give credit. There was another blog post that I think it came up the next day by Alex Bradley at Harvard. It was on the blog called We Beasties, which is part of the Science Blogs group. And he picked up on everything that I'd missed, basically. I don't think he'd seen mine when he wrote his. His background was chemistry, and so he was able to point out the chemical a priori problems before the experiment was even done. There were very strong chemical reasons to say this is impossible, because the chemists had analyzed. It's true that arsenate in particular, has the same bond structure that phosphate does, but it's a bit bigger, the bonds are a bit longer, and they're much less stable in ester bonds, such as in nucleotides and in DNA. So that if you tried to make even nucleotides, much less DNA, out of arsenate instead of phosphate, they would fall apart within less than a second. And this was quite solidly established in chemistry. So there was every reason to say, no, this result can't be right. Alex Bradley also picked up on, I think, a certain amount of evolutionary issues that I probably should have picked up on, but didn't. In particular, the difficulty of having polymerases that could use this stuff, this hypothetical DNA, as a template.
Because it's bigger than a house.
Yeah.
Well, I think of what the authors actually concluded was not, although they were kind of, I won't say evasive, but they were kind of, didn't spell out the conclusions. But based on the amount of arsenate they found in their gel slices, that would suggest that maybe something like 4% of the phosphates in the DNA had been replaced by arsenates. And that immediately raises a whole lot of problems. One is, replacing 4% isn't going to solve any problems. If your problem is that you don't have enough phosphate, replacing 4% of the phosphates in your DNA with arsenic is not going to solve the problem. Mm-hmm. And it creates a second problem. Even if the arsenate bonds were stable, they're not the same size as the phosphates. And that means that the backbone of DNA, instead of being even homogeneous, is going to be variable. And the polymerases will have a very hard time replicating even a polymerase that was sloppy enough to use arsenate. If the template strand is uneven, the spacings between the nucleotides are sometimes longer, sometimes shorter, then the polymerase is going to have a very hard time getting the right bases in. Accuracy is going to go way down. Polymerase will probably just stop working. And if nothing else, the PCR reaction that was used to amplify the DNA for the sequencing shouldn't have worked if there was arsenate in the DNA backbone.
That's a very good point.
Because they did use, the DNA they used was from the arsenate-grown cells.
Also, there are other enzymes that make the precursors. Wouldn't they also?
Lots of enzymes.
Right, and so they would have trouble recognizing the arsenate as well. Yeah.
Even the authors suggested when they were aware of the stability problem, but they said, well, maybe the DNA is, protected by being packaged with particular proteins or by being sequestered maybe in those vesicles we see, which are actually polyhydroxybutyrate granules. But anyway, But this is another red flag. If that was the case, then when the DNA was purified, as they did, purified the proteins away, it should have been unstable, but it wasn't.
Right.
So at some point you decided to work on this strain yourself, Rosie?
Yeah, well, it took about 6 months for the paper. The paper was originally published in Science Express, their advanced online publication, and it took 6 months for the paper. It took 2 days for the controversy to rise up and a couple of weeks for it to die down. But it took 6 months for the paper to actually appear in print, along with 8 critical technical comments and a response from the authors that basically said, well, we could be right about this, and we could be right about this, and we could be right about that.
Let me tell you how I felt about it. It seemed to me that they were talking about everything except the main, simple, straightforward issues of the analysis. They were bringing in all kinds of fancy physics. They were bringing in... It reminded me of the following. Suppose you are looking at a painting and you have to evaluate if it's authentic or not. And you find out that the frame is totally authentic. You do the in exquisite detail, you find out that it fits the time and fits the techniques. And you don't even look at the painting itself.
Right.
So it's that sort of a thing. Their answer had to do with tangential things. It did not deal with the main thing that you and others brought up. So I felt that this was sort of trying to get out of it by shifting the subject of conversation, by changing the subject in a way.
And so I had thought about, there had been discussion on blogs about and articles, whether somebody should try and replicate this work. And the consensus very sensibly was, well, you'd never get a grant to do this.
This is
because
true.
Given the prior reasons to think this result was impossible, which are very, very strong, and the very poor quality of the data, there was no reason to think that result was true. And no sensible granting agency would ever give you money. It would be scientifically a waste of time. But at the same time, there was a strong sense among a lot of people that, to be fair, somebody ought to try to repeat it. And so I decided to do it, partly because of that consensus that somebody ought to, partly because I thought it would be kind of fun, but largely because I would do it openly on my blog. One of the things that had always been unusual about my blog was that I was writing openly about the experiments that I was doing before they were published. So I was just writing almost like a lab diary. Here's what I'm trying to do today. I'm designing this experiment. Okay, what should I do? Oh, here's some results. Gee, I wonder what they mean. Oh, here's a problem I'm just starting to work on. And I was doing this because I thought it important that science be much more, that the process of science be made much more open, so that the general public could get a sense of what doing science is really like, of which the embarrassing truth is that most scientists spend most of their time trying to figure out why their experiments won't work. And I was a very good example of that. But hardly anybody read my blog. So it was a good effort, but it wasn't very successful. And I thought I could do this arsenic experiment in the open, as open science, and it would get a lot more attention than the stuff that I'd been doing, and would help to kind of spread the word that, yes, you don't need to keep your science secret. You can talk about it while you're doing it. So what I planned to do was to try to grow the cells, to purify DNA properly from them. I figured if there's one thing I know how to do, it's to purify DNA, because I study natural transformation and purify DNA all the time. And then I would need collaborators, because I don't know how to do mass spectrometry. I have no idea. I would need somebody who knew about mass spectrometry or whatever was appropriate, who could assay the DNA to see if there was actually arsenate in there. So as is my way, I wrote a blog post saying, I might do this experiment. If I was going to do it, this is what I would do, blah, blah, blah. But I would need a collaborator. And I got an email from Leonid Krugliak at Princeton saying that he and Josh Rabinowitz, I think mainly Josh, had a system that would be excellent for doing this kind of analysis. And maybe we could work together. So I said, oh, great, okay. So, and at that time, the authors had said, because the paper had appeared in print, the authors had said, yes, they were making the strain available to anybody who wanted it. So I sent for the strain, spent a month trying to get the bacteria to grow, eventually discovering that, and I suspect it indeed was that they don't really like glucose, but they grew fine if I gave them glutamate. So I gave them glucose and glutamate, trying to replicate as well as I could the original growth conditions. And then I spent a long time trying to get them to grow reproducibly in arsenate. Some of the time you could give them 40 millimolar arsenate and they were just fine. And other times they all died. And it took me a while to get a rough idea of what that problem was. I think it was mostly getting used to the arsenate, just a short-term metabolic adaptation to it, not an evolutionary sense adaptation to it. And eventually I had purified DNA from cells grown with lots of phosphate and no arsenate, and with lots of arsenate and no phosphate. And I sent it off to the guys at Princeton. And I have to particularly give credit to Marshall Reeves. He's the grad student at Princeton on the project. He did all the analyses, and he's the lead author on the paper that we wrote about it.
And all along, you were documenting this on your blog, right?
Mm-hmm. Yes.
Because I remember at one point, Carl Zimmer wrote on his blog, you have to go read Rosie's experiments. And that must have gotten you huge traffic.
It was at the point where I was tearing my hair out trying to figure how come they wouldn't grow reproducibly on arsenate. And I was writing about the precipice of irreproducible results. And yeah. But people were watching my blog, and it had a lot more readers than it used to have.
I'm sure now
which
you
was
have.
Great.
I'm sure now you have a good steady readership, right?
A modest steady readership. But I have to admit that, somebody else's research project is not that interesting on a day-to-day basis. Mm-hmm. So I'm not surprised that people are not sort of obsessively following whether we can figure out why this particular mutant doesn't take up DNA.
Yeah. No, but you can imagine, though, someone could visit time to time to get a flavor of what research is like, and maybe you would inspire other people to do similar things. I tried this myself a few years ago on my blog. I had been doing some experiments, and I wrote about them. And what I found was that I got great suggestions from people who were reading it, and I thought that was unusual because you go outside your own lab and your own environment, and you can really get a different view.
Yeah.
But I stopped doing experiments, and then I didn't feel it was right to write about what people in my lab were doing, so I stopped doing that.
That's sort of, I refer to people in my own lab kind of as the postdoc, the new grad student, the research associate, not by name, and I generally don't write much about their experiments. Right. I think for me, the blogging about my experiments works really well because for me, it's where I actually do my best thinking about what I'm doing. The act of finding the words to write it in, not too technical of language, for me, really clarifies my thinking. So whenever I'm thinking, I should do this experiment. Oh, Christ, what controls should I use? I'll say, write a blog post. And I'll write a blog post about the experiment that I'm planning. But it's not planned when I start writing the blog post. But by the time I've finished writing the blog post, it's quite well planned. And I've thought about all the controls and et cetera. And the same when I have a result that I don't understand. I'll write a blog post explaining what I don't understand. And by the time I finish that, it's all much clearer in my mind. So in many ways, I'm doing this just for myself.
Yeah, I think it's great. I feel the same way. I feel that writing really helps you clarify your thinking. And I bet a lot of other people feel the same way.
And I find that for some reason, writing on my blog is much easier. Writing papers is a killer for me. Mm-hmm. But writing on my blog, it just comes out.
And it's infinitely more fun, isn't it?
It is, yes.
I love this conversation, but we left a little bit things up in the air about the results that you got.
Oh yeah.
So let's make sure we go back to that. We can return to the idea of what blogs are good for. I think it's a great discussion. But let's talk about the results for a minute.
Okay, the results were very simple because it was a very simple experiment. The bacteria grew proportionate to how much phosphate I added, independent of how much arsenate was added. And we made nice growth curves using this lovely little machine called a bioscreen incubator. The DNA that was extracted from the bacteria contained only baseline amounts of arsenic, the same levels of arsenic that were sort of found in the water that was used as a control for the mass spec, once the DNA had been washed even more thoroughly by the grad student, Marshall. And none of the arsenate was in nucleotides when the DNA was digested.
So you had no arseno-diester bonds.
If the arseno-diester bonds were unstable, we would have arseno-mononucleotides, which were not found. Or if they were subject to normal digestion. If they were somehow both stable and not cleaved by normal DNA's enzymes, then we would have had arseno-dinucleotides, which we didn't have either. Also, I did another experiment. I did the experiment kind of without realizing that was what I was doing, in that I made that when I purified the DNA from the cell, I ran it in a gel to check its quality before I sent it off to Marshall to analyze. And 2 months later, somebody said, if that DNA had arsenic in it, it should have started breaking down once it was purified. So what I did was I took the same DNAs that had been sitting in my fridge for 2 months, and I ran them again. And they were still perfectly intact. So that also says the DNA is stable. And that's actually, if we accept the chemistry prediction that arsenic bonds should break down once the DNA is purified, even if they didn't beforehand, then that's actually a very sensitive test of how much arsenic there could be in the DNA. So basically, we've pushed the upper limit of how much. We can't prove there's absolutely zero arsenate in the DNA, but we've pushed the limit way down. Say there's no detectable arsenate in the DNA.
You can also do this experiment with oligonucleotide machines, because you could effectively use the chemist to make you the arsenonucleotides instead of the phospho-based nucleotides. You could have arsenonucleotides, and you could simply test formally whether or not first you can create such an oligonucleotide, and then you could simply ask the question, would Taq polymerase, using a straightforward PCR, be able to extend product, even using normal nucleotides as your substrate as opposed to arseno-based nucleotides?
Wouldn't that be a bit expensive, though?
It would be, but
and you'd have to be a chemist.
You'd have to be a chemist, but I think the way Rosie went about doing this is extremely elegant because it's all in vivo. The in vitro approach, I think, goes back to Elio's comment, where you don't want to tap dance around the main point. Was the arsenic atom substituting for the phosphorus atom in the DNA?
And the problem is, as far as I know, you can't really make an I guess you'd want an arsenate at the alpha position and then phosphates on the beta and gamma positions of your nucleotides. This is hard chemistry. It's possible.
But also, to take their point of view, just for a second, to take their point of view, they would certainly say, well, a test tube is not a cell. And what's going on inside a cell is different because these bonds are protected in some fashion. That is formally true. So in a way, you can't really kill the argument by doing the in vitro work only. It has to go along with in vivo work as well, I think.
Yeah, I think the sort of, as you politely say, elegance of my experiment came largely from a sense of my constraints and limitations, that I didn't have a lot of time to invest in this. I didn't want to have to learn a lot of new things. I was trying to design the simplest experiment that would get to the heart of their claim. And I think that's pretty much what we did. Anyway, so to finish the story, we wrote up the paper, we submitted it to Science, and at the same time, we posted the paper on the archive site that the physicists use, the sort of preprint repository that the physicists use, where anybody could read our manuscript, the version that we submitted to Science. And we asked blog readers to provide any comments that they wanted on the blog after reading the manuscript. And we got some comments.
Let me ask you a question, Rosie. Science is okay with that? They're okay with doing that?
One of the reasons I chose the archive was because I wanted to post the manuscript. It was part of doing all of this openly.
Yeah, but this doesn't jeopardize your ability to publish in Science, does it?
Right. So, and the reason I was going to just put it like on my website, but then I thought, who knows how fussy Science is about things like that. But the advantage of the archive is that the physicists all use it. And I was pretty sure Science publishes physics papers, and I was pretty sure that Science had to deal in the past with physicists posting papers on archive before or when they submitted them to Science. And so I checked with the editor at Science to say, is this okay? And she said, oh yes, Science has no problem with the archive.
Okay. That's good to know. I had no clue.
Yeah.
It's good to know. So I posted the manuscript, and then Science sent it out for review, got 2 reviews, and then they sent it out for the 3rd review because of issues that they felt had come up. So we got 3 reviews, which were reasonably positive. They were, going into picky details, but basically quite positive. And so the paper's been provisionally accepted. Science wanted us to do one more experiment. We hadn't measured how much phosphate was actually in my medium that I made. I had gathered from how poorly the cells grew in my medium that there was very little phosphate in my basic medium. And I had to add about 3 micromolar phosphate to get the same level of growth that the authors had seen in their medium when they didn't add any phosphate. But we know that their medium was contaminated with 3 or 4 micromolar phosphate. But we had not, I took that, the growth I saw when I added phosphate as indirect evidence of how much phosphate was in my medium. I thought maybe 1 micromolar. But Science wanted us to actually do an analysis of how much phosphate was in our medium. It turned out that was relatively complicated to do. Marshall couldn't do it on the equipment they had, but he found somebody in geosciences who could help him with this analysis. Showed that my basal medium only had 0.5 micromolar phosphate. Now, one of the things that's puzzling is how come their medium had so much phosphate in it?
It's where the water comes from.
Well, I don't know. I mean, I used the same glass distilled water I've been using for 20 years. My expectation was that it was probably because they used, reagent grade chemicals instead of analytical grade or ultra pure, although they hadn't actually specified what kind of chemicals they used. I just used straight off the shelf, reagent grade whatever we had, some of it 20
But remember, a lot of phosphate comes in from how you wash your dishes.
It's true.
And very few people acid wash their glassware in microbiology labs.
No, and I certainly didn't either.
And so you can get phosphate from how your glassware was washed.
From the detergent, right? From the detergent.
From the detergent, that's correct.
Depending on how large a volume you're putting into how large a container, surface area, etc. So I still don't know where all their phosphate came from. It might have been from not using acid washed glassware or from one particular reagent that was contaminated. We have no idea. Anyway, so we did that. We got that analysis. We polished up the paper. We sent it back to Science a couple of weeks ago now. And we're still waiting to hear from them. I'm checking my email constantly to hear from Science for what we expect will be acceptance because we did everything the editor asked us to do. But
well, I expect that Science is, once burned, twice. What is it? What's the saying?
Once burned, twice shy. I think Science.
Twice shy. They must be extraordinarily weary of making another mistake.
They may have sent it out for review again. They just want to be extra careful now.
Well, that's great. I in fact have the manuscript right in front of me. I downloaded it.
Oh, right, and that's the revised version that's on the archive now.
And It's great that you can do that. I've heard that physicists do that, but I'm not aware of. And maybe you're setting a precedent. Did you get good? Did you get good comments from anyone?
We got some good comments. One in particular about the statistical, the error bars. That when you only had 2 replicates, it wasn't really useful to have error bars. You could just show both values, and so that was changed in the resubmission. There was, though, what's funny is that there are other places where things can be posted. Like there was a Nature Proceedings, I think it's called, which was meant to be a sort of a pre-submission repository site. But Nature's just announced they're closing it down because nobody used it. It's just not caught on in biology for some reason. I think biologists are much more worried about being scooped.
Sure.
But one of the things I liked about blogging was that everybody knew what I'd done, and if somebody else come up and said, well, I got my paper in first, doesn't matter, because it's clear from my blog what I was doing and when I was doing it.
That's a good point. I noticed on your blog that you also posted the reviewers' comments.
Yes.
Is that okay to do?
The reviewers are anonymous.
I don't see why not. I mean, they're anonymous.
Yeah, of course.
They're anonymous. I don't know who these people are. They're not sent in confidence to the... There's nothing... When you get the reviews, there's nothing saying the reviews are in confidence.
Yeah.
So, and certainly science hasn't complained. So, yeah, I'd never seen anyone do that before.
No,
but I was then inspired by a snark... I do get snarky comments on the blog, and somebody had posted a snarky comment, I bet you don't do that with the harsh reviews. And it just so happened that a couple of weeks later, we got some quite harsh reviews on a manuscript, sufficiently harsh that the manuscript was rejected. And so I then posted those reviews on the blog, too, mostly because I could let off steam by replying to those comments on the blog. I couldn't reply to them to the editor because the paper was rejected.
That's a good point. I might try that someday.
It just felt good to write it all out there, even though...
Wait a minute, Rosie. I'm not clear. You were just saying talking about a different paper.
Yes, this was a different paper that has nothing to do with the arsenic story.
Just wanted to make sure. Yeah. So, have you heard anything from the other side?
No, not at all. I haven't initiated contact. There have been, situations where other people have suggested that Dr. Wolf-Simon should contact me, and it's certainly. They should, and I made it clear when they sent the strain that I was going to be describing what I was doing on my blog. So presumably they know what I'm doing and have chosen not to contact me. One of the things that I find quite disappointing about the whole story is that the first author, Felisa Wolf-Simon, who is a postdoctoral fellow, has basically been abandoned by all the other authors. She's, a junior scientist. She's in a very difficult position. And none of the other authors are speaking up at all in the past, year and 4 months since the paper was published. There's been not a peep from anybody except the senior author, the last author, the guy in whose lab she was working, who had initially said, we stand by this research, and then more recently was quoted as saying, well, if something gets peer-reviewed, we might have to eat crow. But none of the other authors have said a thing. And I think that's not very good behavior on their part.
Everybody volunteers for a science authorship, but when it gets ugly...
Yeah, suddenly they're treating it like it was contract work, and the authorship was what they were paid to do, whatever. Because a lot of these authors, I think, were brought in because they had particular high-tech expertise to do the different high-tech analyses. And they seem to be treating it as if it was, work for hire and the payment was a science paper.
Yeah.
But no responsibility for the rest of the paper themselves.
That's a problem, isn't it, with multi-authored papers?
Well, I want to talk a little bit about the last question on the science. You had one of your reviewers of your work asked about why such a high level of glutamate, and when I was reading it, when I read that comment, I was really intrigued, and I had this deja vu nightmare of back to when I was a graduate student. I worked on selenite, and I was in a lab that had worked on phosphate. And so I had to learn all sorts of strange things studying for my qualifying exam. So here's my working hypothesis of why the extra glutamate.
Mm-hmm.
Is the extra glutamate or glutamic acid required to fill the energy deficit that's created by the arsenate, which is then substituting for phosphate
No.
in the first electro-level phosphorylation and glycolysis?
No.
Such that the arsenate in a low phosphate medium, like you had in the your low medium, will deplete because the phosphate's going to be put into stuff or scarfed by the enzymes.
No.
She's pretty clear about that.
Your answer is is no. So, well,
okay, let me tell you why no, and then you can go on with your lovely chemical scheme. Because the bacteria wouldn't grow in the basal medium with no added phosphate, or with loads of phosphate, even when I didn't add any arsenic. So before I started messing around with arsenic, I first wanted to get the bacteria just growing in phosphate medium with lots of phosphate. And they wouldn't grow until I added some glutamate. Or they would have grown, they would grow with various amino acids. In fact, what they did, this was a nice little piece of detective work. They would form colonies when I used the medium to make agar plates. The bacteria would make tiny colonies, but they wouldn't grow at all in broth. And I wondered if maybe there was some contaminant in the agar that they were using as a nutrient. And so I made the same plates with agarose. And now the bacteria wouldn't grow at all. So that says there was something in the agarose that they needed. And then what I did is that gave me an assay to figure out what they needed. I made agarose plates, spread the bacteria on them, and then spotted on different test materials to see which materials would let the bacteria grow. And they grew great if I gave them yeast extract or tryptone or various other things I had sitting around on the lab shelf that had amino acids in them. And then I tested glutamate and they grew that. So it was nothing to do with the arsenic as far as I know.
Okay, so it's likely that the anaphylaxis pools are being depleted, and it may actually just need the glutamic acid to refill the pool, or something in the Krebs cycle is stealing it out, so you don't have enough.
Yeah, and I didn't even test other amino acids, but certainly tryptone and yeast extract were great. It wasn't that glutamate worked and the other amino acids didn't work. It was that the simplest combination I had tested happened to have glutamate and a couple of other things in it. And I said, let's try glutamate. And it worked. And I didn't want
Could it be something as simple as the fact that it may be an autotroph for glutamate? The bug being an autotroph for glutamate. In other words, it can't make glutamate.
Well, it's possible, but I think that the sort of complex metabolic explanations are quite likely, because there probably isn't much glucose at all in the sediment of Mono Lake, but there probably are sort of amino acids and stuff. But I didn't want to get it. I didn't want to have to understand the chemistry of these bacteria, especially after I found a very detailed review article about the metabolism and growth properties of Halomonas species generally, and discovered that they weren't fussy. They were very generalist bacteria. They could tolerate a wide range of pHs and temperatures and salinities and nutrients. So all I wanted to do was to find something really simple that would let them grow. And I don't know why they did grow for the authors without any added glutamate, but there are so many holes in the paper anyway that it didn't seem productive to try and track down any particular
I wonder if I imagine that Vincent is looking at his clock, and if that's the case, I wonder if we shouldn't give you a chance to sort of think broadly about where you think this is going and what you think the its proper destination is. What should happen to this whole theme?
Well, so the scientific story is probably going to be a footnote in the sort of sociology of science. The result was wrong. It's something that science journalists are finding very interesting, They won't let the story die. And I think there will be, it would be excellent meat for a thesis, a PhD thesis in scientific
Ethics.
communication or something. Scientific ethics. But scientifically, I think it's just about over. Scientifically, it was pretty much over, within a week. But all of the other implications it has lots of implications for public understanding of science, how science is communicated, how scientists communicate with each other, how new media, Twitter and blogs, which are what I use, are or are not changing how science is communicated. One really useful comparison that you guys will probably remember is the cold fusion story. This is something that a science communication specialist named Marie-Claire Shanahan has been studying. She's at the University of Alberta. How scientists communicated when the announcement of the discovery of cold fusion was first made, and how, again, scientists very quickly demolished the whole idea. Even though they didn't have blogs and they didn't have Twitter, they had phones, they had fax machines.
It just takes longer.
It didn't take a lot longer to do that.
That's interesting. I was just going to comment earlier that this happened remarkably quickly, maybe because of the media, but under the right circumstances, I suppose it could happen quickly as well.
I think what slows down the response isn't so much the availability of ways to communicate fast as how much media coverage it gets to draw people's attention to it. The more of a splash you make, the more people are going to look and say, wait, hang on a minute.
Well, I think in this sense, it's important that your paper is published to put an end to this story. If the other one just stayed in the literature and the scientists knew it was wrong, but the public didn't, I think that would be a problem.
Yeah.
So now that yours will be published, we'll have this, maybe this podcast will come out at pretty much at the same time that your paper is published, which would be great. And the press should see it and listen and say, okay, that's the end of the story. We hope. So the press is guilty sometimes about reporting the initial finding, but then when it's corrected, they often do not go back and fix it.
The press on this, I actually was very impressed with the quality of the media coverage once the concerns started appearing on Twitter. I mean, I put my blog post up on Saturday afternoon, and by Monday morning, I'm being interviewed by a CBC News science writer. And Tuesday morning, Carl Zimmer's story goes up. And everybody who wrote about it really wrote very well. It was ignored by lots of things, and the original announcement got far more publicity than the concerns. But the concerns did get a lot of coverage given that they were saying, oh no, that flashy discovery is wrong. They did get a lot of coverage, and it was very good coverage.
Be interesting to see how your paper is covered, right?
It will.
That's going to be curious. Anything else before we wrap this episode up, gentlemen?
Nope.
Are we finished? I think that's a great story. I think the science is interesting, and I also think the way you're communicating your science is really interesting too. Is Elio and I are big fans of using new media, and
I know I read your stuff too.
This is just another, and you've taken it in a different direction, which is great. And I'll let you know, I read your blog. I think it's really great, even though it's not my work. I still feel.
Did you see my new posts about the paper in Nature about mutation rates? And the one I really liked that didn't get as much splash as I hoped it would have, which was about how to behave at conferences.
I read that
love
too.
That. I'm actually giving that blog post to Amy Chang of ASM, who deals with the students as they approach the big meeting, the big general meeting of the American Society for Microbiology.
Right, which is coming up next month.
I think it's great.
Okay, I'm glad to hear that.
Yeah, it's really good. No, the idea that a gene would have a different mutation rate from another, I mean, how that would work is really puzzling. Right?
If you say so, yes.
Right.
I saw that.
But it was a good paper. Yeah. It wasn't a bad paper. And I've actually had an email from the first author on the paper putting forward sort of his ideas about ways that it could happen. I have to email him back and ask if I can post his email on the blog because it was a good response. And there's been a fair bit of discussion about it on the blog.
Right. Well, keep it up. It's
okay,
great.
Thanks a lot.
I just have one final question for you. Did you get a grant to do this work, or did you fund it from the back of the envelope?
Oh, no, I didn't even try. I couldn't ask anyone else in the lab to work on this. And I like having projects to do at the bench. So I did it myself. And what were the expenses? I had to buy a few chemicals. I had to buy arsenic and a few sort of vitamins and maybe tungsten. I got the trace elements free from a grad student across the road who does bioengineering. So it didn't cost much. And one of the nice things about the Canadian granting system, you don't get nearly as big grants as you can get out of NIH usually, but they're quite flexible about what you do with their money once you've got it. And so I didn't have any qualms about using my transformation research money to study arsenic bacteria a little bit.
Of course, I think that's the way it should be done.
Yeah.
Use money to explore new avenues. And here in the U.S., the NIH has no problem with it.
Yeah. Oh, that's good.
Yeah. Oh, you'll be able to find this episode of TWiM at the usual places on iTunes and at microbeworld.org/TWiM. And if you have questions, you can send them in to twim@twiv.tv, and we can forward them on to Rosie as well, or you can go over to her blog and check that out, and we'll post links to that in the show notes, which you can find at microbeworld.org/TWiM. Rosie Redfield is at the University of British Columbia. Thank you so much for joining us today, Rosie.
Thanks a lot. This is fun.
It's
been a lot of fun. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, what a pleasure.
It was fun, wasn't it?
It sure was. Different.
It's always good to do something a little different now and then.
All right.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Another fun adventure.
Always in the small microbe world.
Well said.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for their technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.